Okay. Can you see my presentation and can you hear my voice? Okay.
Very well.
Okay.
Very well.
Thanks a lot. Well, thank you very much for the invitation to participate in this event. I'm very sorry not to be able to be there in person, but I really want to congratulate Andrea for his new role, and I really want to wish the Centro Fermi the success and the visibility it deserves. Building on previous presentations, today I'm going to try and tell you a fairly complex story of a series of studies that we performed over the last 5 years to try and bring some of the concepts of network theory, particularly as applied to brain networks, into clinical applications. Previous speakers have introduced the functional connectivity, so I do not need to spend many words on this. What I'd like at this point and what I need to point out, though, is that functional connectivity and the organization of functional connectivity into coherent networks is not a privilege of the human species. It's something that is also observed in other animals, like laboratory animals and generally speaking in mammals. So this is an interesting observation because it points to functional connectivity networks to reflect some general principles of functional organization of the brain, but also points to the fact that it can be used as an endpoint in preclinical studies to try and translate findings between human and laboratory animals. Functional connectivity has been systematically scrutinized in brain disease for over 2 decades now. Again, systematically, robust changes or differences in functional connectivity have been observed in neuropsychiatric patients in a number of different conditions. This includes schizophrenia and autism that have been already mentioned in previous talks, but also attention deficit disorders and addiction. To the point that the word connectopathy has been christened and sometimes used to indicate these diseases, pointing to the question whether there is a relationship between beyond mere association between alterations in these brain networks and the actual development of the symptoms. So these are the questions that have been nagging me for years now. These are intriguing observations, but can we conclude that these alterations in functional connectivity are disease specific? Are they robust enough to be used as diagnostic markers or prognostic markers? Do they play a causal role? Are they strongly related with the disease to the point that alterations in functional connectivity can be considered the causes of some of the symptoms of these diseases? And finally, and most importantly, if we tweak functional connectivity, can we actually change behavior? Can we actually have an effect on some of these symptoms so that they can be used to develop new therapies? Now, we understand little about the connection between functional connectivity and disease. And a relatively new tool that's been on the market for a while now, and that's been developed by also some of you who are sitting in this audience today, is network theory, and particularly complex network theory, that has brought about a number of concepts that are particularly useful and evoke some thinking about the meaning of functional connectivity in the human brain. These include, for example, and this is most of our interest, the study of community structure, namely the strongest association between certain clusters of nodes within the brain with respect to other nodes, thus creating what's sometimes referred to as a community structure and modular structure. The reason why this is interesting is that it alludes to the principle of functional segregation and integration in the brain, which is somehow an intuition that neurocognitive scientists sometimes use to describe brain functioning. But that, however, it's very hard to put into more quantitative and into more rigorous spaces. Network theory lends a hand and some insight into this by providing some basis, some mathematical basis to put these concepts into action. Okay. We were lucky enough to be able to pull together a large European consortium some 5-6 years ago, focusing on neuroimaging as applied to the study of alcohol use disorder, which is a form of addiction, and particularly focusing on the use of network theory to try and develop some therapeutic targets. Why alcohol use disorder? Alcohol use disorder is an addiction-related disease which has a very high prevalence. It's an unmet medical need. There is no cure for it. The rate of relapse in patients who are detoxified is close to 90%, and symptoms can be ameliorated but not entirely eliminated. It is an addictive or an addiction-related disease because it shows the same pattern and involves the same brain circuits as addiction to other substances like heroin, cocaine, etc. Its high prevalence is explained by the fact that, unlike many of those other substances, alcohol is perfectly legal and widely available. So it is a particularly difficult condition to combat. I always like to bring up these slides when talking about this kind of studies. This is Nora Volkov. She has been the director of the National Institute of Drug Abuse in the United States. And I like her definition of addiction, and particularly alcohol dependence, as a disease of free will. It really points to the heart of what addiction is, namely the seeking and the consumption of substances against the negative consequences that sometimes are dramatic or even deadly of these substances. This also points to the complexity of this disease, which is very apparent when we look at neuroimaging studies. The number of different circuits that seem to be involved in addiction is wide and complex. There is the involvement of the reward system, which is a very ancient part of our brain, but there is also an involvement of the attention system, of the memory system. Insofar the memory of drugs gets stuck to the brain for many years, even after detoxification. Where this memory is actually hidden is still a mystery, and it is really at the heart of this condition. Needless to say, the disruption of functional connectivity has been observed in a number of studies in alcohol use disorder. Common finding is that there is an overall reduction in functional connectivity, but the questions I pointed to at the beginning of my talk are very valid in this context and have not found an answer. So let me tell you a little bit about our attempt to make some progress in this direction. Our attempt was based on the observation or on the conclusion that just performing human studies, observational human studies in patients is not really sufficient to address these questions. These are very often observational studies in which we compare patients with healthy subjects. we can tell that there are differences in brain connectivity, which are perhaps associated with some of the symptoms, but we cannot tell whether these are predisposing factors or whether they are the effects of alcohol, for example. And we cannot definitely tell whether tweaking these abnormal connectivity can actually have a therapeutic effect. So we undertook a complex translational approach within this large consortium, and we tried and make some studies in humans to try and nail down which parts of these networks are particularly involved in alcohol dependence. We tried to reproduce these observations in animal models to try and figure out whether they are predisposing factors or whether they are the effects of alcohol consumption. And then we used some sophisticated neurobiological tools to try and change connectivity and see whether this can change the pattern of behavior. Finally, we have attempted to bring some of the findings and the understanding that comes from this process into the clinic by performing a clinical trial to test the hypothesis that tweaking functional connectivity can actually have a therapeutic effect. Let's start from the beginning. I'll be very quick going over the various studies, so I will not dwell into the technical details. So bear with me. We performed a clinical study in alcoholic subjects, comparing their functional connectivity with that of matched healthy subjects. We used a network representation of functional connectivity, creating a fairly large network of over 600 nodes in the brain. We found, consistently with many previous studies, that functional connectivity in patients is overall reduced, and that's true everywhere in the brain. The question is whether this decrease in functional connectivity is somehow driven by particular networks, particular areas, or is it just a widespread phenomenon? One way of looking at it is by measuring modularity. In this particular case, we used an infomap approach to break down the network into a set of modules. Strikingly, we found that the modular structure in the brains of alcoholics is pretty much intact, with the exception of two areas, two modules. One is the reward system, the basal module, which contains the key areas of the brain reward system. And this is not surprising, given the nature of the condition we are looking into. And the other area is the insular cortex, which is part of the cortex, which is hidden under the temporal cortex, and is very important for interoception. So it is what perceives and conveys to the rest of the brain the bodily effects of the drug, of alcohol in this specific case. Particularly, we observe a breakdown with the anterior insula changing substantially its connectivity. Okay, this is an observation which is somewhat difficult to interpret. However, the interesting observation that prompted us to go deeper into this is that upon treatment with naltrexone, which is a drug that is commonly used in alcoholics, and by prolonging their period of abstinence by weeks, we had no effect on the breakdown of the basal brain modules, but we observed a recovery of the connectivity of the insula, which is very interesting because it tells us that that functional connectivity, or these alterations in functional connectivity, are dynamic. It can be changed, and the changes seem to somehow correlate with the symptoms. They seem to ameliorate when the symptoms ameliorate. To understand a little bit better what that may mean in practical terms, we looked at the role of the particular nodes within the network. Some of the nodes following Sporns, Bulmore, and the classification by Guimerin and Amaral, some of these highly connected nodes in the brain are deeply set into the modules, so they are responsible for the functional segregation of certain areas. Some others are very important to maintain and ensure connectivity over the entire network. They connect mostly different modules. So they are very important for the integration of these connectivity networks. This is captured by a couple of parameters within community degree and participation coefficient. Again, I will not dwell into the details, but I'd like to point you to the participation coefficient, particularly, which gives us an idea of the diversity of the connectivity of a node towards other modules, and hence its centrality and its role in the integration of the entire network. Particularly nodes that are characterized by high participation coefficients are also very important for the network integration, and sometimes they are referred to as connector hubs. Now, so it happens that if we look at this particular parameter, participation coefficient, that's actually pretty much unaffected throughout the brain, with the exception of the insula. Here I'm comparing the centrality of the insula and of every node in the network in the two groups, patients and controls. Particularly, we have a strong increase of centrality of the anterior insula in the patients compared to healthy controls. This is very interesting, and it has attracted the attention of neurobiologists and cognitive neuroscientists, because that's the way they represent the role of the insula in their cartoons. The insula is described functionally as a hub that somehow conveys the interoception, so the perception of the effects of drugs into other circuits, particularly the ones that are involved in the emotional processing and in decision. So our results were very appealing to them because they indicate that the insula seems to play a much exaggerated role in conveying this information, and it has a much exaggerated integrative role within this network. Interestingly, we observed that the participation coefficient and the role of these of the anterior insula become closer to that of a connector hub in patients compared in controls. And actually, after treatment, it is almost normalized. It seems to go back to normal. Okay, good enough. That's all we could do in humans. This is still observational. We cannot really demonstrate that it has a causal role and that can be somehow used to try and cure the disease. Let me quickly show you what we did in animals to persuade ourselves that a similar effect can be observed in laboratory animals. This is an interesting rat model in which rats that normally hate alcohol and don't drink it because they dislike it profoundly, they're exposed to alcohol vapor, and just by breathing alcohol for a short while, they become strongly addicted to alcohol. So that's an indication of how addictive this substance can be. Again, functional MRI studies in these animals, mutatis mutandis, of course, given the different scale of the brains we are looking at, show exactly the same effect with a strong reduction in connectivity between the anterior and the posterior insula, the separation of the anterior insula, and the reduction in its centrality. So alcohol seems to do to the brains of animals the same thing that it does to the brains of humans, which is interesting and gives us a tool, because in animals we can actually use some neurobiological approaches to try and change the activity and the connectivity of the insula to see whether that can change their behavior. Particularly, we used an approach which is called chemogenetics. Very shortly, one can express some receptors, artificial receptors in specific parts of the brain by viral transfection. And one can activate or deactivate these receptors by administering a non-endogenous substance, which is called CNO. In other words, after we have transfected our rats, we can switch on or off, or we can excite or inhibit specific parts of the brain. So we designed this clever experiment in which we built these switches into the anterior insula. So we can overexcite the insula, or we can inhibit the insula at will, and we can look at how it affects brain activity and connectivity overall, and how it affects behavior. These animals were made addicted to alcohol, so we can actually observe the effects of tweaking the insular activity and connectivity on their patterns in seeking and consuming alcohol. Okay, functional connectivity and functional MRI demonstrated that changing the activity of the anterior insula actually affects all the regions which are within these networks, in which we have represented these circuits, which is reassuring. So we have got two independent confirmations, one coming from resting state functional connectivity and one from selective excitation and functional MRI of the anterior insula. What is most interesting is that exciting the insula did not have any effect on alcohol consumption. So the animals were exposed to alcohol and the alcohol intake was measured. This is a control group in which no receptors were expressed, but if not some GFP. And this is the group in which we inhibited the insula, and this is the group in which we excited the insula. The green group showed a strong reduction in alcohol consumption, but it did not show any other effect in any behavioral parameter that we looked at, like locomotion, consumption of food, or consumption of water. So this is very interesting because it had no effect on what we call natural reinforcers. It only had effects on alcohol consumption. So it seems to be very specific to the addictive mechanisms of alcohol. Alcohol per se is also a nutrient. So we were very wary of the fact that we might be altering the patterns of food consumption, but it didn't. So it looks a very specific mechanism to addiction. And it is surprising that by looking at what by definition is a distributed system, like a network of connectivity, we could actually point down to an area like the anterior insula, like a hub that is actually orchestrating the properties of this entire network, which is involved in addiction. So this was very interesting and suggestive of the fact that the insula may actually be a therapeutic target. Obviously, we cannot perform viral transfection into the insula of patients, but there are other means that can be used to figure out whether changing the insular activity or connectivity can actually have a therapeutic effect. This technique is called transcranial magnetic stimulation. It is a non-invasive technique that uses the Faraday law, basically, to stimulate or to inhibit the electrical activity of neurons in some specific brain area. So what our consortium did was to develop a particular device that could actually stimulate the anterior insula, and we applied this to a set of patients to see whether the stimulation of the insula, like in our rats, could produce a positive effect on the disease. Obviously, this is a long shot, and clinical trials are complex and affected by many parameters. So the conclusive step of this story has good news and bad news and prospects for the future. Let me start with the good news. By looking at functional connectivity in these subjects, stimulation by TMS did actually somewhat normalize the connectivity of the insula and its modular organization. So it looks like this particular mechanism may be useful. The bad news is that when looking at the clinical data, and particularly at the consumption of alcohol, and the consumption of alcohol 60 days after detoxification, we did not observe a significant clinical amelioration. Unfortunately, the clinical data in the sham group, so the control group that was not treated with TMS, overlapped with those treated with TMS. So we cannot conclusively tell that this mechanism has a clinical effect. There are a number of reasons why that could happen. We might not have been able to stimulate properly the insula, which is very deep in the brain and not easily reached. Also, there are lots of confounding factors. The placebo effect can be very strong in an open label study like the one that was performed. So again, our first clinical attempt to bring this long and interesting story into the clinic was not successful, but there is a lot we learned, and we'll definitely proceed on with other follow-up studies to try and figure it out. Okay, interesting. I hope you enjoyed seeing like somewhat mathematical concepts that many of you contributed to discover and to develop have actually been taken so far into the clinic to the point of guiding a clinical trial. So let me thank you for your attention, and let me thank the contributors to these studies, which are numerous, particularly Cécile Bourdier for the analysis of clinical data in drinkers, in alcoholics, Carlo Nicolini for the implementation of the development of the algorithms that we used for the calculation of modularity and centrality, Giulia Forcellini, who participated in the analysis of the TMS study, Giulia Scupa and Stefano Tambalo, who performed all the studies in animal models. And of course, the large consortium that has worked on this for the last 5 years. The clinical studies were conducted at Linköping University in Sweden and in Germany, in Mainz, particularly by Wolfgang Sommer. And again, thank you for your attention.
Thank you, Angelo, for the nice talk. Is there time for some questions? So please, Stefano.
Hi, Angelo. Nice talk.
Hi there.
I have two questions. One is in the first interpretation that the changes in connectivity are the consequence of the alcohol or the behavior changes. Let me say it differently. We know that the subject changes behavior, then this is the addition. And the addition is supposed to have a correspondence aware in the brain with changes in functional connectivity too. So this is your interpretation, that the alcohol is inducing, addition is inducing a change in connectivity. But the other possibility is that the alcohol per se, as a drug, is inducing changes in connectivity. The second question is about the TMS. TMS was effective maybe just because it was rising the activity of the neurons, and therefore the probability of having functional connectivity increased too. So the question is, did you check this effect by stimulating another area not involved, other areas not involved, to see it in the same patient subject if the TMS has a similar effect in functional connectivity?
Okay, these are very interesting and valid questions, and difficult ones to answer. The first one about our conclusions regarding the role of alcohol in functional connectivity. We studied different strains of rats. There are rats that have been selected to be predisposed to alcohol consumption. They are called alcohol-preferring rats. And there are rats that are not predisposed to drink alcohol, that were exposed to alcohol. So we observed the strongest effect in the group that was not predisposed to drinking alcohol, but was made addicted to alcohol by exposure to alcohol. So our conclusion was that most likely these changes in functional connectivity of the insula were somehow determined by exposure to alcohol. Whether they reflect the direct chemistry, neurochemistry of alcohol, or some higher somehow effect related to strictly to addiction, obviously we do not know just from these studies. Again, the fact that changing the activity and connectivity of the insula by chemogenetics selectively affected the addictive behaviors to alcohol would point in the direction of this being more representative of alcohol addiction than just exposure to alcohol. But based on our data alone, I cannot give you obviously a conclusive answer. This is a very deep question to answer. The other one is somewhat easier because we stimulated other areas of the brain, particularly the medial prefrontal cortex, that has entirely different effects on the brain, but it did not show any effect on the insula. So we are fairly confident that we did manage to somewhat stimulate the insula, but we do not know whether the results we are seeing are related to a stimulation of the anterior insula or perhaps the surrounding areas like the temporal cortex. Again, the insula is very difficult to reach with TMS. So we are fairly confident that it's not enough to stimulate any part of the brain to affect the insular connectivity, but you need to be a bit more specific than that. And again, that's all we can tell based on our data. I hope this answers your question.
Let me clarify that for the first part, I was intending to have the addiction per se to be a cause. So if this is true, also the addition of another substance should have the same effect. So this maybe was
okay.
Side effect of the question.
Okay, again, also to that I perhaps can give you a partial but perhaps interesting answer. Obviously, we did not look systematically at all possible substances, but we did look at other substances. Particularly, we looked at heroin, and we did see effects of heroin, addiction to heroin on the insula. We also observed something very similar with cocaine. Pulling this data together would actually indicate that the insula may be involved, generally speaking, in substance addiction and not necessarily only to alcohol addiction. We don't know the neurochemical mechanisms whereby or different substances may end up affecting the connectivity of the same brain region, of course. But it looks like the insula may be involved in the addictive behavior in general, so not necessarily specific to alcohol.